Okay, so like I said off microphone, I do not know exactly how this is organized anymore, so we might go from small to big, back to small, but I will cover each slide as it is. So there's essentially two different cells in the nervous system, and while this is a gross oversimplification, and anybody that studies glial cells will be extremely honked off because glial cells have an enormous amount of diversity, we are going to lump all of them together, and you need to remember that they are the supporting cells. The main stars, the ones that get all the attention, are the neurons. They're the ones who have the most physiologically relevant action potentials, transmit signals via those, and chemicals secreted from the cell and received by the next one called neurotransmitters. I will try to fold interesting factoids into this presentation. How many of you have ever heard the expression, you only use 10% of your brain? Almost all, if not. Where does this come from? Not from some evolutionarily misguided attempt to have way more neurons which suck up lots of energy than you actually need, but rather because 10% of your brain is the neurons, and 90% is the supporting glial cells. Therefore, somebody somewhere, some journalist said, you only use 10% of your brain. Extremely not true. In fact, the metabolic functions of the glial cells that support that are equally important. But you know that neurons are the ones who transmit the signal, so that's great. We will remind ourselves of the histology of that, but only briefly, of which one is a neuron, which one's a glial cell, remembering that the neuron is the big one. We will have you draw the distinction between a neuron and a nerve is simply a bundling of the fibers that come off of a neuron. So it's a collection of axons, essentially. Okay. We'll move on. So, like I said, I want you to understand the hierarchy of the central nervous system. There are many ways to parse it, and while we may not specifically quiz you on each one of these different titles, they are all relevant to your understanding how things are organized. So here they are. Central nervous system. One of the major, well, really what we're actually going to cover in the most detail. The peripheral nervous system probably has about an equal number of neurons as does your central nervous system. So in a sense, if you think with your gut, you honestly might be able to do that if you all link them up the same way you did the central nervous system. But those that are not connected to the spinal cord of the brain are the peripheral nervous system, and that involves anything that's responsible in collecting information about where you're at. So if your skin is telling you you're hot or something's got some pressure on you, that's part of the peripheral nervous system. If you're involved in transmitting that signal from the central source, so getting from the spinal cord out to the muscles, that's also peripheral nervous system. That's called the somatic part of it. And then if you actually need to modulate the activity of your guts or other smaller components, that's here in the autonomic nervous system. And I've put in these keywords here to help me remember exactly how this breaks down. But if you need to motivate the body to either get ready for fight or flight, that's the sympathetic part of the autonomic nervous system. It's something you're not... John, get off your phone. A Skype warning just came up for John, poor John. Or get your roommates off your computer. And parasympathetics for the resting state. So, not as important to what we're going to test you on, but definitely there. Okay, so the central nervous system, the spinal cord, off on its own. The brain has many different parts reflecting many different stages of evolution. We're probably pretty proud of our mammalian brains, and that's really just one part. If you look at the brain, it's going to be layer upon layer, essentially the more exterior layers being evolved later. So we'll parse those. The more primitive stuff, of course, connected most directly to the spinal cord, so the hindbrain, in order moving up No, not in order at all. In backwards order, comes up from the spinal cord, hits the medulla, hits the pons, hits the cerebellum. That's essentially the hindbrain. You can ignore this term. The midbrain, we'll spend one brief moment on. It's involved in motivation. And the forebrain's where you do most of your thinking. Some of these terms aren't great, but note the presence of the main thinking structure, the cerebrum, the famous wrinkly part on the outside for us, the limbic system, which is, again, not a great term, but it's kind of the next layer in, something like the hippocampus. Then the kind of the central junction, intersection of all things brain, the thalamus, that helps communicate between the different regions, and then the part that interacts with the endocrine system, the hypothalamus. Okay. All these things come into play. Your job is to identify them and know their functions. Okay, spinal cord is actually fairly straightforward. We've got some wonderful figures in here for you for all the different parts you're going to need to be able to label. Then we'll look at the histology of it again and remind you. I will tell you at the outset you need to be able to tell your top from your bottom, and the histology will make that clear. So we'll look at that again. But notice that the white matter is on the outside, the dark matter is on the inside, the gray matter rather. Remember that the white matter reflects the presence of the myelin sheaths, the insulating glial cells that are helping conduct the electric signal from the anteriormost in the brain down to where the signal needs to go, the various places where that motor action can happen, whether it's a limb or part of the guts or whatever you're communicating to. It's about transmitting that long-term signal. The other things we're going to bust you on, there's some wrappings on the outside that's called the meninges. Sometimes it's the dura or pia mater. You might see that term. Okay, and then one of the reasons you need to know the top from the bottom is because we're going to make you distinguish between what is transmitting information back to the central nervous system, that's called afferent, and what's taking the effect from the central nervous system back out to make your motion, for example. So that's through motors, your motor neurons, and your efferent system. For that, it is very helpful to spell very well and use your English properly. I am with an A affected by sad things. Just like the afferent nervous system takes in sensory perception. I'm cold. Those signals go in the top, they go back up to the brain, get processed. Whereas if I want to effect a change, I want to make a change, that's the efferent system. It's on the bottom, the ventral side, and that's where the motor neurons and the motor ganglia come out. Okay. There is one feature that I didn't mention in there, the dorsal root ganglion. That contains afferent sensory information, and it's a more dorsal structure. Just spend some time with this slide. It gives you a general sense of the organization. This slide, this figure here in particular, shows you some of the more relevant things. But here it's time to point out, here is this ventral cleft. Ventral, the bottom. In our case, toward our belly, where our back is the other side, the dorsal side. So this is our very back. This is toward the belly on the inside. This is where the motor neurons, the efferent stuff happens. This is where the signal processing stuff happens, the afferent. And there's some finer details there for your information. So here it is in histology. I put this note in here. Observe the direction in which fibers go. You're not just here to learn vocabulary. Hopefully you could sit here and marvel at the wonder that is mammalian or vertebrate biology, our engineering. So one of the things that's actually interesting to look at is how is one thing plugged into the other, and the same is true in the brain. But you'll notice various means with which one side communicates to the other. One of them is through this juncture up here. You'll see most of the nerves in cross-section, and that'll be the little white dots. That's the axon surrounded by the myelin, yet again. Those will be on either side because this is going to the brain, this is coming back from the brain. And I'm sorry I get lost in thought so often, guys, but remember, you can identify the ventral side by this cleft right here. Notice that the two sides are jammed together, so it may not be clear as day, but it can definitely be distinguished under the microscope. Okay. One more thing is with the fibers going out. Notice we talked about those dorsal root ganglia, the sensory ones out here. I'm not going to sit here and advocate that you can actually identify that on this slide. In my opinion, this is probably the muscle right here, but I wouldn't swear to it. But you can definitely see that the nerve fibers have actually started going outward. So this happens periodically as you go down the spinal column. You know, there's different things to communicate to at different levels along the AP axis, and you're going to have these different dorsal root ganglia coming in and different motor ganglia coming out down here on the bottom. Okay. So you can see the direction of the nerves go this way. One point of terminology that I forgot, ganglion is just a cluster of nerve bodies. Okay, so that if you get a group of them together and they're not involved in any other bigger structure, or if they're very distinct from another structure, they call that a ganglion. Okay, so here's a human model of a human brain and the very roughest kind of structures that you need to learn from it. The names are intuitive. The frontal is in the front. The temporal is next to your temples. I think occipital has something to do with being Western, which doesn't necessarily make sense, but that's in the back, and you can memorize parietals on top. The primitive brain connected to the spinal cord is down here, brainstem, cerebellum, famously responsible for motor coordination. We'll get into all the different structures when we look at, and their roles as we look at the sheep brain. Notice that sheep are not quite as smart as we are. So this structure here, the cerebrum, which is the outermost layer of the newest part of the brain, is not very big, but you still have those characteristic bumps and valleys, the gyri and sulci. So you can identify that as a homologous structure to our nice wrinkly brains, and eventually we're going to cut through this sagittally right along the middle and expose all this stuff on the inside. Yeah. And so there are two different slides of this. I think this one, I'm not going to dwell on this one, I'm going to dwell on the next one. But this one has a bit cleaner images. You can see some of the detail a little bit better, the colors a little bit better. But for your purposes, I've crossed out these terms that I think have nothing to do with what we want you to learn. But all this other stuff is pretty relevant. I may have crossed out pineal gland too, but another hormonal signaling center. But yeah, I'll give you the grand tour on the other slide. Here we go. Hopefully it won't let me forget anything in particular, though I already see that it will. So let's try to check off all the different parts and all the different functions. Cerebral cortex is this thick layer out here wrapping almost everything and what we traditionally think of as a brain. That's higher functions, memory, thinking, complicated tasks. Ali? Ali says occipital comes from essentially the ocular root, so dealing with the eyes. And now we can all remember that too. And the next class, unfortunately, we'll be dealing with these podcasts as well, so they'll be stuck. But thank you. Do not confuse cerebrum with cerebellum, very similarly sounding. But again, higher function is here. Remember, brain sheep don't think that much, therefore theirs is much smaller. If you're really concerned with how superior humans are because of our brain power, there's two things that got inflated. It's the cerebrum, and that's the biggest one that we think of, and the cerebellum. And why did the cerebellum become so inflated? Can we conjecture on this a little bit? Thoughts?
Control an opposable thumb.
To control an opposable thumb? Well, let me put it this way. This is my grand thinking point. What is the defining characteristic of hominids? Talk about it in your groups real quick. We've simmered down and our answers are table 1. Walking upright. Table 2 agrees. Table 3 agrees. And now, of course, table 4 does too. But I don't. At least when I was growing up, they told us, oh, the thing that makes us so great is we can work with tools and we have opposable thumbs. But none of that really mattered until you could actually stand on your hind legs to actually use your hands for something. So one of the major things that drives all of our evolutionary goofiness is the fact that we walk upright. And they think, well, some of the hypotheses for this are, you need a big-ass cerebellum to be able to do this. Walking on 2 legs is an inherently wobbly thing. Okay? And all the different little adjustments that you have to do when you rotate on your ankles is very unstable structures. And just to be able to rock like this, ever walk behind somebody? We're at a Catholic institution, you ever walk behind somebody as you're going up to get your communion? It's funny, you look at the whole line and it's every other person wobbles. Maybe you can do it at the theme park too, I don't know. But you realize that walking is a very unstable thing. So we need a big cerebellum to control for this. That's what its major function is. Write it down, we're going to test you on. There, that got you to break the spell. All right. I think one of the things that drives our intelligence then is then not only do we have the ability to use our hands to use tools, make opposable thumbs and so forth, use our opposable thumbs, but also just getting that thing so giganto-sized drives a bigger brain in general. Not necessarily true for the poor ostrich, but it's a hypothesis. Okay, I tried to make that interesting. Let's move on through the other stuff. One thing that's missing, I'll tell you at the outset, this hole is called a ventricle. It contains cerebrospinal fluid. Its homeostasis is important to general functions of the brain. You should know that it's there. You should know what it's called. Much like in the spinal cord, there is a series of fibers dedicated to transmitting information from one side to the other on the interior, and that's this corpus callosum. It's extremely white matter because it's all insulated for getting this signal across the sides of the brain. I think now let's work from the back up. Spinal cord obviously is responsible for communicating to the rest of the body. There's also some reflexes in there. And essentially I raised the point of evolution of these brains. If you're simpler chordate or working your way up through amphibians and then the lower mammals and so forth, you keep adding layers to this brain. And the most primitive, essentially, that we have much in common with stops here at the midbrain. But I want you to think, as you work up the length of the hindbrain, midbrain, forebrain, the further you get, the more advanced the feature, the more advanced the function is. So medulla oblongata contains fight or flight stuff, controls the pressure on your arteries, it motivates heart rate and stuff like that. And let's see, pons gives you some of the sensory input. A lot of different things are plugged into this from cranial nerves. It's also essentially the highway for communication from the cerebral cortex. The cerebral cortex is another name for the cerebrum, essentially. The pons connects that with the cerebellum because there is balance and motor information involved in the cortex as well. Then once you get to the midbrain, you're actually talking about a part of the brain you need for kind of advanced behaviors. It's the motivation center, but there's not too much thinking going on. But if you're interested in feeding or mating or all the various things you need to do to survive, this is probably the structure that's motivating this. Again, there's a another great TED talk about, I think it's called the Golden Circle, about how the different parts of your brain, the ones that evolve first, tend to dominate your decisions and work outward. But sorry, I digress. If anybody's interested, I'll let you know. I'll tell you one little story about the midbrain. Remember that its function is to give us our motivating behaviors. Let's stick with mating and eating. Anybody else think of anything else that feels really good, that if you're a rat in a cage you would keep pressing that button? Sorry, what? Food's definitely in there. Reward. What's a reward? What things stimulate rewards? What things don't happen on a Friday night at a Catholic university? We're too shy to say, but essentially we're talking about sex, drugs, and food. Okay, anything that's pleasurable. And okay, so this is essentially the dopaminergic pathway, and you may have heard about this because it's the one that goes haywire in Parkinson's. So what I want to reveal to you is, okay, yes, you can put these pieces together. Parkinson's, dopaminergic, pleasure, people taking drugs. You mix all these things together. What you can do is abuse the system, and it starts not knowing how to inhibit itself, and ultimately gets damaged. So narcotics use can be one of the ways to get to a Parkinson's disease state. Okay, it's not necessarily the only way, for sure. But then think about what the symptoms of Parkinson's are, and it's principally motor control. Okay, your hands jitter, you can't really control your muscles that well. And what I want to let that illustrate to you is that all the different parts of the brain tend to have some kind of interaction with the different functions. So even though the midbrain is the pleasure center, the motivation center, but it's still tied into the motor things that are going on in the cerebellum, in the cerebrum, and the more motor-related stuff, more reflexive stuff that happens in the hindbrain. So all these things tend to overlap, so it gets confusing. Okay. There's really only one more thing to point out here. I'll get to the nerves in a second. But that's the thalamus. Thalamus is what's left over from the diencephalon during development. You remember that from our chicks? Maybe. But crud, I told you it's confusing. Now I've forgotten the thalamus. Oh, right. This is basically the integration of everything. If you're going to have a thought in the cerebral cortex, it's probably going to be processed through the hypothalamus. It's the crossroads. And if it needs to be sent along to the motor effectors or for processing for balance, it's going to go in the cerebellum. It's going to go through the thalamus. Importantly, this is the structure right above the hypothalamus. Okay, hypothalamus is not that great for to identify based on just seeing it on these guys, but it's an important piece of your brain. It's the one that ties in with the hormonal regulation of the body. So the hypothalamus is, according to its name, right below the thalamus, and that's what actually translates nerve signals into, hey, we're afraid, hey, we're aggressive, hey, we need to increase insulin, hey, we need to make our boobs make milk, hey, we need to, by the same mechanism, get rid of that baby in our belly, and so forth. So lots of things that are controlled mysteriously, even making your kidney work, come through signaling from the hypothalamus. Hopefully that covered all of that, but yeah, okay. This is taken strictly from Wikipedia because when I had to relearn this last year to tell you anything about it, these were the most useful descriptions to me. I already mentioned this, but I think it deserves special mention, that fight or flight happens in the medulla. So, sorry, let me start over. When I look at this text and I read it and it's very opaque, the medulla contains the cardiac, respiratory, vomiting, and vasomotor centers. It deals with autonomic, involuntary functions such as breathing, heart rate, and blood pressure. Now, how many of you caught that they said that's each of the same thing twice? Okay? Or almost. Cardiac, heart rate. Respiratory, breathing. Vomiting, not so much. Vasomotor, blood pressure. Okay? All these things have to do with taking flight if you want to get out of someplace in a hurry. You need your blood pressure to go up. You need to breathe faster. You probably don't want to vomit, but notice that it's not a particularly high function. It's not particularly high up the brain. But when you're reading these things, do stop and see if you understand what they're actually saying. So we're not going to get you too bad on this, but it's something we want you to observe while you have the sheep brains in hand. There are 12 named nerves that kind of fly around on the outside of the brain, kind of like the keyboard wire on the computers that you guys don't use anymore, and the webcam cord and the monitor cord and all this is flying around your actual computer. That's the way I think of it. But when you plug them all in nicely, you get a nice functioning system. These are their names. A lot of them have to do with moving the eye or something attached to the head. They're essentially nature's way of improvising a way to move that funny new addition that is a head. Remember, the vertebrate head really is a new invention based on the neural crest. But right. Now I know why I did this. This is, we tossed out that term parasympathetic system at the beginning. This is a diagram showing the cranial nerves coming into some of those structures. So here in the medulla, there's 7. There's 2 branches that actually that come into the medulla, 9 and 10. So I'm not going to get them all perfectly correct, but 10 is definitely the vagus nerve, which has to do with getting the heart to beat. Remember, fight or flight said we're going to increase the heart rate. I thought 7 was facial, but essentially what I'd have you do is look at this, look at the corresponding cranial nerve, and say, hey, each of these things are plugged in to a different part of the lower brain, and it kind of makes sense with their function. So you can see some of these nerves on the outside, and I just want you to try to identify as many of them as you can and relate them to the other structures that your book outlines that you should catch. So the olfactory bulbs are probably ripped off of your sheep brain, but they'll have some leftover, just lines that represent the nerve track. The nerves from the eyes come in and meet at the optic chiasm, where they actually switch sides where they go. There's other ones plugging in again to the hindbrain. So if you can see, trigeminal's not really in the medulla. I think it's more closely into the pons. This is one of the ones for the eye to move sideways, I believe. There's one on the underside that makes the eye move up and down, I think. The specific function is not that important, so it's okay if I get it wrong, it's okay if you get it wrong. But try to look for these little appendages coming off, and you'll kind of get a sense of these wires coming off the sides. I should point out at this point, because I forgot, the pons is kind of hard to identify from those photos. It's much easier when you have an intact brain, because it's that part that essentially wraps around on the anterior side and plugs back in to the cerebellum as it goes back to the dorsal. So the dorsal side's hidden here, the top, right? This lump is interior, and it's kind of a C shape that wraps around and plugs into the cerebellum. And like I said, it's much easier to identify whole than when it's been chopped in half, because it kind of looks contiguous with the medulla oblongata. Just a warning. Okay, so we got special sensory systems, eye and ear. I am going to cover those next time. Fair warning, if you want to study beforehand, you need to be able to trace the path of sound to actual perception, and a light wave to actual perception. Pay particular attention to what's actually a clear structure, layers, histology, blah, blah, blah. We'll get into all of it. But there's a lot for that too. Okay, so we will start with the external view of the eye and the conversationally relevant pieces. So there's only the one model for the eye, and then we're going to dissect the sheep eye. But make sure you take it apart, make sure you know which structure is which. And so let's just start. Very obvious things, less important, but there are the muscles that support the movements of the eyes, innervated by various cranial nerves. Again, the order and names of all of which aren't that important, but except to observe them on your brain, which one's going this way. Everybody's favorite missing term when they're talking to somebody, well, what's the white part of the eye? That's the sclera. Okay, good term to know. Why is it white? Because it's primarily connective tissue. In your book, we're going to have these discussions of the different tunics. You should be able to correlate those to the other more general names that we have for them. So this will be the fibrous tunic. Connective tissue makes it hard, therefore it's protective. It's white, therefore it's not transparent, therefore it's not part of any of the optics that actually detect light. One of the major things about the eye is you have to get a certain set of features to be completely transparent to get the light into the eye and detect it. Okay? Speaking of that, there are two major transparent structures. That's the cornea on the outside and the lens on the inside. Don't get this wrong. Okay? I confess, learning this for the first time, teaching this last year, I totally thought the lens was the part on the outside. For those of you with better knowledge, that might seem a little crazy, but the cornea is the tougher part on the outside. If I want to venture a guess, I'd say that's actually part of the fibrous tunic, but it's been clarified. It's the thing that they're checking for with glaucoma when they put that little tuft of air into your eye. If there's not sufficient pressure behind that, that's a sign for glaucoma eye disease. But it's structure number one that's clear, that's going to get most of the light focused onto the eye. So the principal focusing power of the eye is in the cornea. Multiple choice. What is the principal focusing element that collects the most light for the eye? Is it the cornea or is it the lens? The answer is the cornea. Okay? This is the biggest quote-unquote lens in the system. It's collecting most of the light. The lens is, on the other hand, responsible for the fine focus. Okay? When you dissect your sheep eyes, this will be clouded over. Again, with my guess that it's part of the fibrous tunic, but it doesn't have the active living cells clarifying the cell. So the fixing makes it cloud over. I think to another degree, the same is true of the lens. So other things we're very familiar with. The iris is visible here. That's the muscles used to dilate the pupil. Pupil is the hole itself. Yes, you do need to label this on your path of a photon into the eye, the pupil. But that said, it is just the hole. That's the amount of light getting into the eye itself. That's the entire function of the iris. There is a separate set of muscles that control the shape of the lens, and we'll get to those once we're on the inside. Note also that there's vasculature coming up outside of the sclera. Okay. So I said there are two things that are clear, two major structures. That's the cornea on the outside. See, it is contiguous with the sclera. The lens behind that is the oval-shaped guy. This guy's actually squishy. When you dissect him, the fixed one is very hard. That's not true in the living tissue. It's actually very stretchable, and that's done by the choroid on the side here. And when my brain's working, I'll tell you what this ciliary marginal zone. This has a set of muscles that'll stretch the lens. Going back to clear things, the vitreous humor is the squishy jelly on the inside that's been cleared out. If not enucleated, that is, if it hasn't had its nucleus taken out, it's been moved around in a specific way to help the light get through. And these cells are actually lined a very stretched, long line of cells so that the light kind of follows along it like a fiber optic cable. It's actually a really cool structure. What else do we need to know about? You need to know the order of the layers, and the order of the layers makes sense. You got the protective part on the outside, that's fibrous. You got the vascular on the inside, serving what the layer more interior to that, which is the retinal or the neuronal layer. So protection on the outside, the service industry on the inside, and the highly sensitive, doing all the work, neuronal layer on the inside. And the neuronal layer is also called the retina. Okay? Part of that layer is a pigmented epithelium that collects any bouncing around light. So that's just kind of a way to refine. There are two terms here. This will confuse you on the test, so take notes. The macula is the bigger structure, the fovea is the smaller structure. We can boil both of these down to a highly sensitive area that contains most of the photoreceptor cells. The fovea is the smaller area that contains the very strong condensation of color, cone cells, that finally resolve the images that you see and interpret as color. We as primates are very unique to do most of our processing via light, or most of our sensation via light. If you take away our sense of sight, it's a major debility to us. If you're an insect, you're going to see most of the world via flavors, smell. And likewise, most of your interpreting power is going to be dedicated to what you're best at. So for us, it's light. For an insect, it's smell, and they'll essentially see smell in color. And if you're a duck-billed platypus, it's reading electricity in the waves. Most of your brain will be dedicated to interpreting electrical signals in the water. Little points of interest. When you're doing your dissecting, make sure you note the optic nerve coming out the back. The presence of the optic nerve creates a disruption in the retina that results in the blind spot. So this nerve, which is a bundle of all the retinal layers, axons coming out from the photoreceptors. Photoreceptors are a set of neurons. When we bundle all those axons coming out together, they exit in the very back, in the very place where we're trying to do most of our detection, and send the signal back to the brain. But it does disrupt, it does cause a blind spot. You don't detect the blind spot because your brain interprets over it. Anything I'm missing? One other thing that we seem to mention a lot is that there's a set of fluid in the anterior chamber here between the cornea and the lens, and the posterior chamber supporting the lens. We just give that a mention. I don't know if it's a big deal. Another great view. I'm not sure that it contains anything different from what we said before. So we'll move on. Right, so this confirms what I said before. The fovea is actually the subset of the macula. Okay, after this we're going to switch gears to the ear, but I think it's a good time to mention one of the points in your book, and that's to talk about the different kinds of neurons that detect sensations from the external world. It completely amazes me, try to go along with me on this little journey, about how even on the cellular level, form begets function. So if you wanted to detect something quantum mechanic like light, you use quantum mechanics. If you want to detect something mechanical like the disruptions in air that are sound, you use something mechanical. And if you want to detect something chemical like a taste or a smell, you use a chemical detection. And all these get interpreted by the neurons, send on their signals, get processed, and so forth. But I just want to mention to you these different types. Here we go. So we're obviously interested in detecting light. This is going to be the first mention of us detecting color, which I don't know how many of you know, but we detect in 3 different channels: red, blue, and green. And we spend a heck of a lot more of our energy on the green signals and refining those because they're much more of a natural, they're more important to our natural environment. The other colors that we see are combinations of these, and I'll discuss that in detail. But suffice it to say, we have a series of proteins that absorb light. They're called opsins. And when they're absorbed, they can transmit that signal via, what's Einstein? What's the prediction of E equals mc squared? Photoelectric. One of the predictions of the theory of relativity was the photoelectric effect, so that if energy in the form of light hits something material and gets absorbed, it will it'll induce a currency. This is how the photocells on your calculator work, or any photocell works. It's also how your eye works. It's also how photosynthesis works. So the light hitting a material generates a current, and your body harnesses it and sends it on as a signal. That's photoreceptors, so that's quantum mechanics. These guys here, this is a scanning EM of hair cells in the ear. So these detect actual vibrations in the fluid inside your ear caused by the sound that originally hit your eardrum. And those waves in that very minor ocean in your inner ear pull back on these hairs here and physically open the protein that they're attached to, which is connected to the action potential to send it on down. So it's kind of like an old school teapot where it's got the little nozzle over there's the bigger hole at the top, but then there's a little thing that clamps down on top of it that whistles. It's like lifting the top of that with a little lever on it. And when that opens, it allows the ion flux to start an action potential. So again, a very cool mechanism. I should have used that in the description of photoreceptors. And I'll mention one other one. It's a form of chemical detection, and that's going to be important to your taste. So, any idea what this molecule is? Let's see if I can highlight it. Candidates, I've given you a hint on the right. It comes up when you're eating hot wings. What's that? A burp? That is hilarious, but not quite correct. I was way too late getting it. It's capsaicin. So this is this alkaloid molecule, and what's the sensation you get when you eat hot food? 1, 2, 3. It's hot, yeah. Okay, and it has a correlative. What happens when you eat mint? What sensation do you get? Cool. You don't think about it, but it's true. That's the same as if you rub icy hot on you. It has menthol and it has capsaicin in it. Both of these things activate a receptor protein not dedicated to that purpose at all. Not dedicated to food at all. It's dedicated to thermal regulation. The same thing that makes you feel cold, the TRP channels, responds to these molecules. It essentially gets stuck to them and leaves them open and firing. So that when you eat something with capsaicin or rub something with capsaicin on it, you feel hot. And it responds to the same thing with menthol, makes you feel cool. So it's a cool example of chemical detection, and we use it in taste. Except if you actually wanted to taste something or smell something floral or bitter or so forth, there'd be dedicated proteins for that purpose too. So when we get down to the cellular level, there's all these cool things happening. So please have a gee whiz moment somewhere along the line. Go to Wikipedia and have fun. Okay, so photo detection. We do have one exercise on this, but about after images. And I tried my example before, and it's kind of cruddy for our purposes, but I'll send you this file with a good one. Before you do that, you need to be able to do this kind of color math, and I guarantee you it will get you. But here it is. Blue is caused by activation of blue photoreceptors. Red is caused by activation of red photoreceptors. Green is caused by activation of green photoreceptors. Those are easy. The other ones are not intuitive. Yellow, for example, is the combination of red and green. Magenta, purple, is more appropriate. It's the combination of red and blue. Cyan is the combination of blue and green. white light is us perceiving all these different channels at once. So red, blue, and green all activated at the same time. Now the math that we're going to get you on is, if the real thing is your photoreceptors can get tired, and these cone receptors, they're dedicated to a single channel. There's only red in this particular neuron, or there's only green in this particular neuron, and they only have so much opsin. And when you spend all that opsin by staring at something red, or something really bright like the sun, eventually those photoreceptors will deplete their opsins and they'll no longer work. Really, I think the thing is they think they're continuously working, but that's an inversion of logic we're not going to get into. Suffice it to say, if you look at something red for forever, you're no longer going to detect red. Okay? So that's what's goofy. So then, if you burn yourself out on red and you look at something white, and remember, white is the transmission of all these different wavelengths, what will you see? So you subtract red from white, what will you see? Take a second to talk to your peers. Okay, we got it. So what's the answer? Cyan. Cyan, because white minus red equals cyan. Okay? If you burned your eye out on red and you look at something yellow, what would you see? Green. The green would continue getting through to green photoreceptors. The red would be too tired to notice. Okay. So there's a great example of this that you may have seen. I didn't know it was actually Beyonce, but apparently that's the Beyonce of maybe 10 years ago instead of 5. But like I said, I'll give you this example. You take it home and you stare at this picture, then you stare at something white, preferably a wall rather than another computer screen. The trick is, if you're doing it at a distance, there's too much, it's not a concentrated enough part of your retina to actually get the effect. So you need to be very close to it. But just to run through an example, those of you who don't play with Photoshop too much, I have to explain this. Bear with me. Probably some point in your life there'll be some form of design that you'll have to do at some point. So maybe it's not a complete waste. But colors are made up of different mixes of red, green, and blue. That's how the pixels are displayed on your screen. And if you look at something that's bluish like this, odds are it's going to have a lot of blue in it, but it might surprise you what else it has. So this one has plenty of blue. It's at about half strength. It measures from 0 to 255, so maybe a half or 1/3. And the other ones just kind of balance it out a bit. There's a little bit of red, there's a little bit of green. That gives you this color here. Now when you invert that, and you do this math, subtracting 255 from that to get what's the remainder, you would predict that 141 plus 114 gives you 255, so 141 is the inverse of 114 here, and so forth down the line. 161 balances out 94, 196 balances out 59. If you plug that color into the color mixer and hear those numbers here, you get this kind of maroon but kind of fleshy color. And if you actually compared that to what you see up here, it's accurate. Now, if you looked at this image, this inverted image here, then stared at something white, given the appropriate amount of time, it would essentially do this inversion for you. Such and such photoreceptors will be such and such tired that they'd subtract themselves out, and you'll see a lifelike flesh-toned person after that. So we are going to do it on very simple colors, subtracting only green or subtracting only red and so forth. But that's what's going on with that effect. And this is my example that doesn't really work. When I did this, ultimately a cyan and a white screen will appear here. I saw something red on the white, so or orangish, and it's because it's not complete. It's not 100% effective using computer screens, but at least be able to predict what you should be able to see with this. I would predict if you burned out blue on cyan, you should see green. Here you should see yellow. But again, it depends how long you stare too. Okay, onto the ear. remember, we need to trace the path of sound coming in. I see no better way to discuss this than to just go through it. Let's see. So we have a fancy name for the outside of the ear, auricle or pinna. Remember one of them. You're going to go through the auditory canal, hit the tympanic membrane, your eardrum, and then this is connected to the inner ear via some fancy machinery. The three smallest bones in your body are involved, and just an interesting evolutionary point I want to point out to you, this used to be part of your jaw. If you look at a reptile jaw, you'll see 3 extra pieces in there that have become your inner ear. Okay, but moving on, you'll hit the tympanic membrane. That is connected to the malleus or the hammer. That strikes the incus or the anvil, which is connected to the stirrups or the stapes down here. You need to find these on your models. That stapes hits a membrane on the vestibule of the inner ear. That membrane is called the oval window. Almost nobody got it on the test last year. Please, let's fix that this year. The oval window is important for the transmission of sound just as much as the tympanic membrane. Once you're in the vestibule of the inner ear, the place to go for sound is the cochlea. Multiple choice. Where is sound detected? Is it the cochlea, the vestibule, or the semicircular canals? Please, 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 please tell me cochlea for sound. That's this snail shell shaped coiled structure that contains a specific set of hair cells for detecting sound. That is not to be confused, but understandably confused, with the hair cells in the vestibule, in the utricle, composed of the utricle and the saccule, which are important for detecting linear acceleration. Okay, I'm going to use the other view to talk about the other roles of the inner ear, but suffice to end here that sound is detected in the cochlea, and it goes out through the nerve for that. Look in your books for the fancy name. So, vestibulocochlear nerve. There you go. It actually, therefore, probably serves both that and orientation. So, job number 2 of the inner ear is determine how your body is situated. We've all spun around and made ourselves dizzy. We've all sat in a car and felt acceleration. And so this is the apparatus that detects that. And as with the hair cells in the cochlea, the hair cells in the vestibule are detecting the movement of fluid in that cavity. So there is a distinction to be made here between linear acceleration and horizontal or angular acceleration. So linear acceleration, when you're in your car and you put on the gas, that's detected here in the vestibule. Okay? And it makes sense. Your body's pointed in this direction. You have some movement. Inertia moves the fluid against that surface, and you get the action potential in those hair cells. Likewise, it makes sense that these semicircular canals, one for each orthogonal direction. So there's one, if you rotate, if you bow your head over to the left and right, if you bow your head back, and if you spin as if you were to make yourself dizzy. There's one of those that responds to each of these. So if you want to make yourself dizzy, you go on this one. If you want to nod your head forward and backward, do this one. If you want to go move your head sideways, you do this one. Those I think we can safely lump and make multiple choice answer to them be the rotational acceleration. But that's to be distinguished from the linear acceleration down here. I won't get you on whether it's horizontal or planar or whatever, because I can't tell you which is which myself. Okay? But there are 2 different functions there. They are all giving you information about where your head is relative to your body. Also use this view to pick up the shape of the different bones in here and get their order correct. The hammer hits the anvil, hits the stirrups, and the stirrups look like stirrups. I think that's it. There's one more slide just showing you about the scale of these bones. I may tack on one additional slide on this to trace the path of light, but it should be fairly simple. It's going to go through the cornea first, go through the lens, through the vitreous fluid, onto the photoreceptors in the back, including the macula and fovea, transmitted via the optic nerve, and then interpreted by your brain.